Hi Mahesh, Parthi here. I thought giving a general overview of what we have been discussing on the rare diseases through this video. This may be a better way of communication to begin with, because the purpose of this way of communicating with you is that we want to ensure that our end goals, the end objective of this whole task that we are aligned with. So we have been talking about rare diseases. So we want to work on the rare diseases. Um, Rare diseases. This is what we have been talking about. And we have an understanding that rare diseases are not rare. And our understanding is that there are about 7,000 rare diseases, and these are all we already know about it. And so the way that rare diseases, which are defined in US, is that any disease which is affecting less than 200,000 people. So, assuming that the US population is somewhere around 320 million, so this amounts to somewhere about 1,600 uh, people. One person, if he gets affected out of 1,600, so this is considered as rare disease. Whereas in Europe, this is relatively little different number, that is 1 in 2,000 people, one person out of 2,000 who gets affected, then that disease is typically considered as rare disease. And of course, then there is the 80% of the rare diseases, 80% of rare diseases are genetic in nature. So which means that is kind of a bit chronic. And 75% of these rare diseases are during the childbirth. It affects the infants and so on and so forth. So this is our the kind of entire landscape of rare diseases. So against this background, we have been talking about what is it that we can do it in the knowledge driven approaches. When we say knowledge driven approaches, first let's take one step back and let's look at the how the pharma industry has evolved. So basically, first in vivo, so which means that doing experiment outside the glass vessel, that is in the human body. So inside the animals or inside the human. Then there is a in vitro. So in vitro meaning inside the glass vessel, that is in the laboratory test. So this is how we try to understand the mechanisms of the disease, and we devise new methodologies for treating the diseases through in vitro. And then in the last 30 40 years, the new approach, which is in silico, that is doing the experiment in computers. So we fall somewhere in the area of in silico. But again, the in silico is some kind of very abstract terminology, everything which is related to computers and so on. But then it's time that the field has expanded so much, we need to differentiate from in typical in silico in a what is happening these days in the pharma industry. Now I'm trying to, for our own convenience, I'm trying to, in silico is typically what happens in silico is a lab scientist, they synthesize some drug compounds in the lab, like typically 100, few hundreds, then pass it on to the biology department, they do the biological testing. Then with these data, that is sent to the in silico team internally, and then they predict which compounds are good as a potential drug in the future, which are not so good. So these are all the kind of a models, simulations, they build it in silico. But in the last 10-15 years, the whole field of in silico has exploded. tsunami of information. So there is, I coined two phrases. One is in litero. So the in litero meaning there is so much of research outcome has come in, and the issue here is, the challenge is that everything is in the form of unstructured form. So we want to get the data out of the massive amount of information that has been accumulated in the last 30-40 years of pharma research. Can we create something based on what has been done so far? So that is in litero. In litero is basically getting the data. First we get the data, then we do the connections among within the whatever that is available. So which means that basically this is where two things which comes in. Number one, which is we can say the descriptive analysis and exploratory analysis. These exploratory analysis, these two things we can kind of bring it, classify. These are all again tentative, so just this is it's not a textbook material. I'm just sharing it based on my interactions and experience and so on. So the descriptive is basically what is there with our data. So we need to understand what do we have, how many records we have, how many columns that we have, what are the features that we have. So those are the things we can say descriptive analysis. So then what is exploratory? Within exploratory, again, it is a kind of analyzing our own data, whether the feature A, how much is it correlated to feature B, A versus B, some kind of a correlation analysis that we do within the existing data. Here we are not trying to predict, or we are not trying to suggest anything. We are just trying to understand our own data with the use of descriptive analysis and the exploratory analysis. In descriptive analysis, we are just trying to take stock of the situation, trying to understand what is the average, what is the standard deviation, what is the lowest number, what is the highest number, how many records that we have, how many columns that we have. So these are all the descriptive stuff. Within this descriptive, we are trying to correlate with one aspect with another aspect, that is exploratory. Connecting the dots as well as that comes under exploratory. Can we make sense out of it? Can we infer something out of that? That is exploratory. Now, the another aspect which I call it as in machino. So there are two things. We are taking a clue from in silico, which is a conventional phrase used in pharma industry, but then that I am trying to divide in litero, which means that literature driven analysis. That is literature meaning whatever the journal article, whatever the patents, whatever the conference abstracts, these are all the literature form, literature information. So that literature information, with the use of that, how many abstracts, how many articles, how many data points, how many proteins, how many inhibitors, these are all the descriptive and the exploratory which falls under in litero. Now what is in machino? Here again there are two things. One is predictive and another is prescriptive. So predictive is that I have a data which are all in the form which I can feed it into the machine. Now the last, maybe I would say after 2017, this is a critical year where technology started changing from conventional modeling simulation to some of the advanced algorithm which got into the pharma industry started accepting, which is the ML or deep learning, or all of them put together. We can call this as AI. So we kind of interchangeably we will use in our whole this rare diseases task for the next 6 months. We may call that as a AI or machine learning or deep learning. We interchangeably we will make use of it when we are conversing. So which all means almost the same, unless we specifically we tell this particular algorithm or that particular approach or this particular framework. Otherwise, we would be making use of these three phrases: artificial intelligence, machine learning, and deep learning in an interchangeable fashion. And these all became started to penetrate the pharma industry, the pharma research, especially the drug discovery research in a solid way. And then it started to give some success stories also, like moving a compound from preclinical to clinical. There are some success stories which are already in the market. So with that component, which I call it as a not only the predictive, but also currently these algorithms are becoming powerful. Still, it is not a matured one, but still they all started to deliver the prescriptive. What is mean by prescriptive? Predictive is essentially I have these are the 1,000 options. Hey machine, can you tell me which is the best one out of these 1,000 options? Predict whether it's good or bad, classify them, or rank those 1,000 options that I have, which one I should go for it. Predict and tell me which is good, which is not so good, which is moderately good, which is active, which is not active, which is inactive, which is moderately active. Tell me some of these things. This is what we call it as a predictive. Whereas in the prescriptive, which is the kind of it is like this is where the human nature comes in. Hey, I understand this whole space. Fine. I'm not expecting you to predict. Tell me what should I do? Dream. Dream for new compound that can solve my disease, that can solve, that will be a potential treatment for a particular disease. That is prescriptive. So descriptive, exploratory, predictive, prescriptive. So each one of them, as we move from descriptive to exploratory, from exploratory to predictive, from predictive to prescriptive, the complexity exponentially increases as we move from descriptive to exploratory to predictive to prescriptive. So this is something which is potentially possible with today's state of the art technology. So can we do this in the space of rare diseases? This is what potentially we can contemplate, discuss about, and come to a consensus. What is it that we want to do it? What is it that we're going to do it? And what is it that we are going to achieve at the end of 6 months? Right. Now, let's move on to the next aspect. So now let's talk about the in litero part. In litero, which I mean by collection of data to build our own knowledge platform, which will have the repository of data from diverse sources. So when we say diverse sources, it is not only the sources are from different places, but also the context. Also, there is a bit of a difference. That is, number one is on the medical, on the other one is on the biomedical, tech, the more of a molecular level. The other one is more at the medicine level. That is from a doctor's perspective and biomedical researcher's perspective. So there are two aspects, and in these two aspects, data are available. And when I say data, especially in the last 5-6 years, the concept in pharma industry called FAIR, FAIR, findable, actionable, or accessible. Findable, accessible, interoperable, and repurposable, reusable. So fair principle. So the data that is collected should follow the certain norms, certain principles called FAIR principles, and which almost all the biomedical researchers they have agreed upon. Now within this, there are, as I said, there are two kinds of information related to rare diseases. One comes from the practitioners, the doctors, from the patient point of view, like how the rare, what are the rare diseases, how to define it, what are the symptoms, what are the causes, how big is the population, how these rare diseases, the various diseases which are connected, how that can be classified from a medical doctor's perspective. The other area of information which is available for the researchers is from the molecular level perspective, which is like what is the mechanism of action? What is that which is changing, the mutating at the genetic level, which is causing certain changes in the phenotype and which is causing the disease to the humans? And what are the potential remedies in terms of at the molecular level, at the what is the kind of a molecular formula that can go and inhibit the certain diseased proteins which is behaving in a weird way. How do I arrest it? Trying to understand the mechanism, what is causing the disease. So all that is from a medical the biomedical researchers purpose. So we need to kind of bring these two sets of information. For example, and in the context of rare diseases, so rare disease, when I say just rare disease, I do see that there are quite a few links which pops up, and these are all the knowledge repository. Say for instance, a list of rare diseases information. So when I look into this takes me into this website, and here all the 7,000 rare diseases which are arranged in an alphabetical order. Say if so every single diseases, and again this is from a medical doctor's perspective. So let us say that if I say Hashimoto encephalopathy, and if I want to look for the information about this rare disease, when I enter that phrase and I look for that particular disease, and what has been recorded in that is that if I choose on this, I can see what are the synonyms for Hashimoto encephalopathy, which is the short form abbreviation HE, and also it has another alternate name, steroid responsive encephalopathy associated with autoimmune thyroiditis. And of late, people seemingly increasingly using this particular abbreviation for Hashimoto encephalopathy, which is SREAT. SREAT, steroid responsive encephalopathy associated with autoimmune thyroiditis. And then there is a general discussion, signs and symptoms, and of course then the causes, affected populations and related disorders, diagnosis, clinical testing and workup, standard therapies, what are the treatment which are available for this disease at this moment, and then investigational therapies which are currently ongoing, and supporting organizations. And there are various references related to this particular disease which we can pull out and then do that. So likewise, for all the 7,000 diseases, the rare diseases for which the information is deposited here, and this source of information comes from US. So then there is, so we can write a script and scrape all the 7,000 diseases from this source, and we can take as one bucket of information. And likewise, the second source, this again comes from US, and this is from NIH, National Institute of Health, and this is more of genetic and rare diseases information center, GARD. And we can browse the information by disease, and here again I can say Hashimoto's encephalopathy, and it pops up, and then I can go inside, and whatever the information that is available, they have captured it in various buckets here: population estimate, symptoms onset, symptoms, cause, specialists, genetic testing, patient organization, resources. And if we keep selecting on this, we can go deep inside, and then we can pull out the information that whichever is that we are looking for. And if the information is not around, then it is hashed here, gray colored, cause, genetic testing, which is no information is available. But then still we have quite a lot of useful information. We can browse these down, and whichever the information that we are looking for, we can kind of scrape these information, and then we can create our own one local bucket. And to begin with, probably we might be interested within this the symptoms and the causes and the rare diseases. So basically, rare diseases, the list of rare diseases, what are the causes and symptoms. This is again more from a medical doctor's perspective, and this resource of information and this one, these are all coming from US. Now there is a new initiative in Europe on these rare diseases and orphan drugs, and combined with the European Bioinformatics Institute, they have collected all the information related to the rare diseases. And very interestingly here, one of the very useful piece of information is the ontology. The medical doctors, a team of medical doctors, they they worked for quite a significant amount of time that they have spent on this, and then created the ontology within the rare diseases. This is not the general diseases, all the diseases. This is only exclusively the rare diseases ontology which they have created, and we can make use of this ontology for in our own knowledge repository, and we can pull out their ontology that has been built by them, and we can bring that ontology inside our knowledge platform. And then for every individual diseases for which the information that we collect from multiple other sources, we should explore the way to append, add those information in a structured fashion, so that it will be searchable within this rare disease ontology. So this all put together is from again a medical doctor's perspective. So this will be more useful from a diagnostics perspective. But now we want to move towards the treatment. So when we are looking at the treatment perspective, probably we may have to go at the molecular level. So what is it causing the disease, or what is the gene which is responsible, which is kind of either point mutation or any kind of a mutation. There are very, very many types of mutation. So what is the type of mutation, and so is it a one gene mutated? So all that information that we need to get into that level. And again, we are looking at the knowledge based driven approach, meaning based on the existing information we want to hypothesize. We wanted to come up with a suggestion. So it is not that we are whatever that we are going to come up with is this is the remedy. So what we are saying is, from the knowledge driven approach, we are trying to come up with a potential solution, potential remedy for any of these diseases. So is it possible to build around on this concepts? That is what the one of the objectives of doing this exercise. Say for instance, now we have an ontology for rare diseases from a medical doctor's perspective. Now I want to look at from the biomedical researcher's perspective. There is a disease ontology which is again existing, which we can accept it, because this particular ontology, which was built by very many researchers, or probably over the last 20 years, at least more than 15 years. So that is the amount of knowledge which is already sitting inside this disease ontology. Now we have a rare disease ontology, and here are the molecular level information which is sitting in this for in every diseases. Can we extract the data of the diseases? For instance, there are 7,000 diseases which are listed in the rare diseases ontology. Here there are probably close to 11,000 entries, 11,000 diseases information in this disease ontology, which includes rare diseases. We don't know how many of them belongs to rare disease. So this is one of the other exercise that we need to do, that is extract the information from the disease ontology related to rare diseases. Can we append that, or can we make it a communication between the rare disease ontology information with the rare diseases information sitting in the diseases ontology? Can we make it two-way communication? So this is one aspect that we need to build it. Now from here, this is not the only thing that we have available in this. Also, there are quite a lot of information which is available for rare diseases. Say for instance, in this particular paper, so this paper is about, it's an excellent paper, the knowledge-based approaches to drug discovery for rare diseases. So in the knowledge-based approaches, they have kind of consolidated what are the kind of information that we have related to rare diseases, and this is one of the very good starting point. So Malakards, Paros, which has all the biological targets associated with the disease, and this is extremely important piece of information. Say for instance, you take any particular disease, as long as we know the mechanism of action, this is the particular protein which is behaving in a weird way and which is resulting in a disease, one of the rare diseases. But this one is to do with all possible diseases, but we are looking at out of 7,000 diseases, how many records that we can find how many rare diseases that we can find from this paras data set, and if we find any of the rare diseases for which the biological protein is reported, then we want to extract that piece of information. So if we can find out that particular biological target associated with that particular rare disease, then we can find a small molecule compound that can go and interact with the biological target. So that is what this particular piece of information will be very helpful. And likewise, there is something called OMIM, collection of both human genes and genetic disorders cross-linked by association. So the OMIM database, which talks about the disease associated with the genes. What are the genes which are kind of having some errors that is resulting in certain diseases? So those are the things that we need to pull it out, and we need to explore all these sites, whichever is we feel that relevant for our purpose. Then we will have to pull out that information. Say for instance, this is OMIM site, an online catalog of human genes and genetic disorders, and we can search for OMIM for clinical features, phenotypes, genes, and more. And there is paras, and here we can enter any Shimoto. So if we have any of those targets, it will give us asthma, let's say. So what are the asthma related? There are 418 entries related to the targets, 24 entries for diseases, and so on. So these are all the paras which brings the information about the biological targets and diseases detail. And there is another very interesting repository, Rare Disease Info Hub, and where searching FDA orphan drugs, what rare diseases do you want to learn more about? Let's say Hashimoto encephalitis. So which is steroid responsive encephalopathy associated with autoimmune thyroiditis, and this goes and fetches information for us from multiple sources as well. So this talks about description and symptoms, communities, providers, and research, financial resources related to this particular disease. So we can enter the disease name and then we can pull out all the related information. Say for instance, these are the researchers who are working in one is in Brighton, Massachusetts, and Vancouver. They are working in this area of disease, the Hashimoto encephalitis, and their PubMed articles are, we can access it. Clinical trials, if there are clinical trials, such information will be there. NIH grants, FDA orphan drugs, and so FDA Orange Book drugs related to this particular disease, and related articles, and Chemotex. This is publicly available web server that mines the published literature in PubMed in the form of medical subject headings, mesh terms. The goal here is that using this link, the database steroid responsive with the autoimmune. So this kind of connecting the various textual information, that is the text-based drug target disease. So this is the relationship which is extremely important, and maybe going forward we need to explore something called graph-based approach. So connecting the various points, can we make use of the graph-based approaches into this? So in these things, the different nodes which talks about the drug, biological target, and what is the remedy, that is the drug component. The drug is the chemistry, target is the biology, disease is the medical stuff. So how do we connect these three dots? So this is what is the important piece of information that will potentially would give us what is it that we are looking for out of this. Now, the another most important one, this is the kind of a bit manual curation concept which comes into it. So here obviously, so we can say Hashimoto encephalopathy, and then we can look for all the journal article. So one way of looking at things is that can we identify some of the mechanism of action for the rare diseases, at least to begin with, some, let's say some 25 rare diseases, 25 or even 50, minimum 25, maximum 50, let's say. So 25 to 50 rare diseases, we will identify, cherry pick from the 7,000. So within this 25 to 50 rare diseases, we will pull out all the relevant articles for those individual diseases. From there, we will start extracting the information from these journal articles. We don't know what are the fields that we need to curate it. We don't know. But meaning we haven't arrived at the conclusion. We probably may not be able to arrive at the conclusion immediately. So, but we need to manually start working on this as a trial basis. So once we achieve that, with that experience, we should be able to build our own guidelines and control vocabulary. And why are we doing this? There are a couple of ways to look at it. Even our own approach that we have been talking about, the knowledge based platform that we want to build it to find a certain potential outcome for these rare diseases, there are two ways that we can look at it. Number one, which is an idealistic way. Ideal way is that we will start pulling out all the information from everywhere, connect the dots, try to build a platform out of it. But actually, our end objective is not just building a platform. Yes, we do. That is our end objective. There is no doubt about it. But then, still, our end objective purpose is, can we find some potential solution for any of these rare diseases through connecting the dots? So, one way to look at it is that the last couple of years, the new concept which is emerging, which is called PROTAC. That is, in our human body, there are very many proteins are there, but then not all the proteins are. So let's look at it this way. Let me go back to my slide stuff. So, if you look at our human body, which has about 30,000 genes or proteins, that we can say that, and as of now, the number of biological targets which the researchers across the world are exploring are somewhere around only 1,000 out of this 30,000. And if we look at the number of targets for which already the drugs are already available in the market, probably would be somewhere around 650. I don't know the exact number, but then it should be somewhere around 650. That is, when we go to a medical shop, pharmacy, whatever the drugs that we are seeing, the thousands of drugs in the market, all of them are targeting somewhere around 650 biological targets in the human body. So, but our human body has 30,000, which means that the rest of them are unexplored yet. So we don't know how many of those unexplored potential biological targets are associated with the rare diseases. We don't know now. So, but then the point is, not all the 30,000 biological targets are druggable. That is number one. Also, not all those 30,000 biological targets are associated with diseases. So, if the target is not associated with the disease, then there is no point in finding a drug or any small molecule which goes and sits inside the packet of those protein. If it is not associated with the disease, no use. That is number one. Number two, yes, this particular protein which is associated with a particular disease, but which is not druggable. There is no binding pocket where I can go and fit my small molecule which goes and sit inside. It is not. So if it sits inside, which is druggable, if it is kind of a slippery surface, even whatever the drug that we are consuming, which cannot go and attach to the protein, sits inside, bind to that, then it is the drug will not do the purpose. So basically, what is happening is there are 3,000 close to possibly which are druggable proteins in our body. Druggable, which means that the small molecule which can go and bind to the packet. And then there are 3000 disease associated protein that we have. Out of this 30,000, 3,000 are associated with one or other diseases. It could be rare disease or general disease, many diseases, and we do not know, we do not still have any remedies for those diseases, but we know that they are all, we are estimate, this is an estimate, the 3000 is an estimate, 3000 targets in our human body which are related to one or other diseases, and 3000 proteins in our body, not necessarily all these 3000 are different. 3000 are having a binding packets, active site, which is druggable. If you want to make a drug which can go and binds to it, 3000 are, so meaning anything that we are trying, we are looking for a treatment that should fall in this area. That is, it should be druggable, but also it should be associated with any of these diseases. So this is roughly, we can say that around 1000. This is the 1000 that we are talking about here. So roughly about 1000 targets which potentially druggable, which is also associated with disease. This is what we are looking at now. But this is what the last 30, 40 or 100 years the research community is working on. It should be druggable, it should be disease associated, and then for which the entire big pharma companies, small pharma companies, startup companies, they are all focusing in this narrow region in our human body. But the one good new paradigm shift which is happening in the last 2 years, or maybe last few years, but actually it is really heating up in the last couple of years, which is, yes, it is disease associated, this entire 3000 space, but then these remaining 2000, they are not druggable, but we know that they are all associated with the disease. Then we don't need to go through this conventional mode. Let's take some rare diseases. Most of those diseases are life threatening. What is the typically a drug does? When you are taking the drug which goes and arrests the protein, right? But if this does not have the active site, we cannot stop the protein to function by blocking it. But the new concept is, this is the protein causing the rare disease or any of these diseases, destroy the protein, not just inhibiting. So if enemy comes here, if an enemy is waging a war, two ways to attack. One is stop the enemy, not to wage a war. Number two, just go and kill him, destroy him. So this new technique, and this is not something new in the human body. As we age, there are good protein and bad protein all the time. It keeps created in our human body. So the natural mechanism is all the time the bad proteins are getting destroyed by our own human body. So when the bad protein starts accumulating, if the mechanism doesn't work properly in our human body, that is not destroying the bad protein, then probably we may end up in getting certain diseases. So this is a natural mechanism. Always there will be a creation of good protein, always there will be a destruction of bad protein. This is by nature, we do have this mechanism in our human body. Can we make use of that destruction mechanism towards the disease making proteins? That is the new concept which is last couple of years which are emerging. So this is, can we design, this is where the one that I was talking about as a prescriptive analytics, can we build a knowledge platform so that we have all the information. So when you work in the information rich environment, then we can move towards predictive and prescriptive. Within the prescriptive, can we design a new treatment or molecule or a new drug that can go and destroy these disease making proteins and bring in a cure for some of those diseases? That is something we may want to contemplate. So, this is the whole perspective that I'm giving it. When we start working towards that, so now this looks very dreamy, it's a concepts are too big, but then we are starting from scraping some of the from the medical perspective, which we have already started from the medical researchers perspective, extract all the rare diseases information, and we are looking at extracting the information from the genetic and symptoms and causes information. Then we are also now started looking at the ontology. So how we are trying to build. So this is how we have already started, and also we have identified somewhat around 30 rare diseases. We started manually say collecting the data related to some 30 diseases. So once we collect all the information, then we will start building our own guidelines and control vocabulary for that. So that will take a couple of more weeks. So once we standardize that protocol, then we can make the people to start curating for all the 7,000 diseases. And in parallel, we will start integrating the data from multiple sources. So we will build a knowledge platform. So the knowledge platform in the sense that collecting the information from diverse sources, and it will be a kind of a standard platform wherein we should be able to identify ideally the biological target, identifying a new biological target for a disease, certain disease, because we still do not know mechanism of action for very many of the rare diseases. Is there a possibility that we can build a knowledge platform by which, by connecting the dots, can we infer certain biological targets for some of these rare diseases? So this is an idealistic picture towards which we want to build a platform, and we still do not know what are the other challenges that may pop up. So as we travel through in this direction, we will start addressing those challenges, and that is one direction. The second direction is the brute force approach, which is by connecting the rare diseases with certain biological target, we can start moving. Within this, there are two directions. Number one, go towards the conventional inhibitors mode. The alternatives go towards destroying the bad protein mode. Can we have a AI framework that we can make use of it and try to design a new drug potentially out of that? So that is the second approach. Let me repeat. Number one is collecting all the information in idealistic mode, which is a long-term goal. The brute force approach is with the existing certain piece of information randomly cherry picking, collect it, and then start moving towards the prescriptive analytics. So these are the two approaches, and within that second approach there is a 2A, 2B. 2A is conventional inhibitors, 2B is destroying the bad protein. So can we build an AI framework for that? So this is what I'm holistically looking at the tasks before our hand. Thank you. Thank you, Magesh.